Picture the last time you were truly alone. Not peaceful alone, the other kind. The kind where a Sunday afternoon stretches into something that feels like static. Where you open your phone for the 5th time in 10 minutes, and there's nothing there. Where the room around you feels somehow smaller than it did before. That feeling has a name. Most people assume they know what it is. They don't. Loneliness doesn't just feel bad. Your brain doesn't register it the way it registers sadness, or boredom, or a difficult week. Something categorically different happens. Something physical. Something you can see on a brain scan. And the longer it goes on, the smaller your brain literally becomes. In 1984, a neuroscientist at Ohio State University named John Cacioppo began asking a question nobody in serious science wanted to touch. What does loneliness actually do to the body? At the time, loneliness was considered a feeling, a personal problem, something to be managed, not studied. Cacioppo disagreed. Over 2 decades of research, first at Ohio State, then the University of Chicago, he built the most comprehensive scientific picture of social isolation ever assembled. What he found upended almost everything researchers assumed about stress and the brain. Lonely people had chronically elevated levels of cortisol, the same hormone your body floods into your bloodstream when a predator enters the room, as though your nervous system was treating social isolation as a survival emergency. Because it was. Your brain was never designed to be alone. For the overwhelming majority of human evolutionary history, being separated from your social group wasn't uncomfortable, it was lethal. No group meant no food, no fire, no protection from predators or the dark. Lone individuals didn't survive long enough to pass on genes that were comfortable with solitude. So your brain built in an alarm. That alarm is loneliness. And like any alarm designed to keep you alive, it is not gentle. It is designed to be impossible to ignore. What Cacioppo realized, and what took the scientific community years to absorb, was that this alarm doesn't just create emotional pain, it creates physiological change. And the longer it rings, the deeper the change goes. In 2016, researchers at MIT led by neuroscientist Kay Tai published a landmark study on socially isolated mice. Not sick mice, not stressed mice, simply mice that had been kept alone. What they found in the brain tissue changed how researchers think about isolation. The prefrontal cortex, where you make decisions, regulate emotion, and stop yourself from doing things you'll regret, was showing measurable changes in its white matter. The myelin sheaths that insulate neurons, that allow signals to travel cleanly and at speed, were thinning. The wiring was degrading. Then there's the hippocampus, the structure where your brain converts experience into lasting memory, and one of the only regions in the adult brain capable of growing new neurons. Chronic stress suppresses this process almost entirely. Loneliness is chronic stress. Measured over years, persistent loneliness correlates with documented reduction in gray matter volume in both the hippocampus and the prefrontal cortex. Not as a tendency, as a difference you can see, with your eyes, on a brain scan. Your brain is, in the most literal sense, smaller. But here's what's fascinating, because it turns loneliness from a state into a trap. Geneticist Steve Cole at UCLA spent a decade studying what isolation does at the molecular level. Lonely people don't just feel more threatened. Their immune systems are literally rewired to expect threat. Inflammatory genes upregulate, antiviral genes downregulate, and that same hypervigilance bleeds into social perception. Lonely brains begin reading neutral faces as subtly hostile. Ambiguous words land as rejections. So the brain does what it does when it perceives danger. It pulls back, becomes more guarded, just enough to make genuine connection harder. And the harder connection becomes, the longer the isolation extends. And the longer the isolation, the louder the alarm. Cacioppo called it the loneliness trap. The very changes loneliness makes to your brain make escaping it measurably more difficult. The alarm that was supposed to save you starts to imprison you. In 2023, the U.S. Surgeon General designated loneliness a public health epidemic because the mortality data demanded it. Chronic loneliness raises the risk of early death to a degree comparable to smoking 15 cigarettes a day. 15. Cacioppo spent the last years of his life trying to understand how a species engineered for togetherness had arrived here. He died in 2018, before the epidemic had a name. What he kept returning to was this: the human brain is not a standalone organ. It is a social organ. It grows in relation to other minds. It deteriorates without them. You are not designed to exist inside a single skull. And the question that stays with you, after the scans, after the cortisol data, after everything, is whether we are already living inside the answer to an experiment nobody chose to run. We don't yet know what that produces, but we can already measure the early results. Curious people don't stop at one question. Neither do we. Subscribe to Default Mind.